Hi, this is Alex from devstories.eu. AlphaFold Protein Structure Prediction, Episode 6 of 21. When you run a protein structure prediction, the neural network is rarely the bottleneck. The real waiting happens before the model even starts, dragging hours out of your day just to search through genetic databases. Predicting structures with ColabFold completely changes this math. The standard pipeline for AlphaFold relies on a tool called Jackhmmer. Jackhmmer scans massive terabyte-sized local databases to find evolutionary relatives of your target protein. This search builds the multiple sequence alignment, or MSA. The MSA is the critical input that tells AlphaFold which parts of the protein are likely to interact based on evolutionary history. However, generating this alignment the traditional way takes hours of compute time and forces you to manage enormous datasets on your own infrastructure. ColabFold modifies this pipeline by targeting that specific bottleneck. It replaces Jackhmmer with a different search algorithm called MMseqs2. MMseqs2 is heavily optimized for high-speed sequence searching, but ColabFold goes a step further than just swapping algorithms. Instead of making you run MMseqs2 and its associated databases on your local machine, ColabFold offloads this step entirely. It takes your input sequence and sends it to a dedicated public MMseqs2 server via an API. This server handles the heavy lifting. It searches its own centrally managed databases and returns the finished MSA directly to your environment. This architectural shift means you do not need local database storage at all. This is exactly why you can run ColabFold effectively within a Jupyter notebook hosted on cloud services like Google Colab or uCloud. The setup requires almost no infrastructure. You open the notebook and paste in your amino acid sequence. This could be a wild-type protein or a custom mutated sequence you're experimenting with. When you run the notebook, the execution happens in two distinct phases. First, your cloud environment makes a fast API call to the remote MMseqs2 server. The server computes the alignment and sends the MSA back to your notebook. This cuts a process that normally takes hours down to mere minutes or even seconds. Now, the second piece of this workflow kicks in. Your notebook passes that retrieved MSA into the AlphaFold neural network. This is where the actual folding prediction happens. Unlike the database search, this inference step runs locally within your cloud instance. It utilizes the GPU attached to your Google Colab or uCloud session to calculate the spatial coordinates of the protein structure. Because the slow database search is outsourced and accelerated, your overall iteration time is drastically reduced. You get the highly accurate neural network prediction you expect from AlphaFold, but you get it fast. If you need to test how a specific point mutation alters the physical shape of a protein, you simply edit a single letter in your sequence variable, run the cell again, and evaluate the new output. Here is the key insight. ColabFold proves that by completely decoupling the data-heavy alignment search from the neural network inference, you can put world-class structural biology tools into a standard web browser. That's all for this one. Thanks for listening, and keep building.